Hey guys, I want to take just a second to review a little bit of our material from last week. So in the last lesson, we talked about mitosis and the cell cycle, and that's really important. It's very basic. All levels of biology, the most basic levels of biology, know about the phases of mitosis. You may have learned those in middle school, actually. I don't know if you did, but a lot of middle schools do learn the phases of mitosis. So it's super simple and you just have to know them. So you just have to commit those phases to memory. So to just review, we have I, PMAT, C. I is interphase. That's the first phase. It has G1, S, and G2. The G stands for growth. The S stands for synthesis. And so we're synthesizing DNA. That's when the DNA is copied. PMAT is the actual mitosis part. So all of this together is called the cell cycle or cell division. But it actually, the mitosis part is really where the cell is actually dividing. And so when we're talking about mitosis, we're specifically talking about the PMAT part, and that's sometimes referred to as the M phase. And then last is cytokinesis, and that's the division of the cytoplasm. So in mitosis, remember, P is prophase. The nucleus is going to break down the chromosomes. The chromatin condenses to form chromosomes. M is metaphase. That's when they line up in the middle. The chromosomes are in the middle. A is anaphase. That's when they pull apart. Okay, and then T is telophase. T stands for 2, so you're starting to see the formation of 2 new cells there, and the nucleuses are going to start to reform in telophase. Okay, and so you just really have to know those phases and what's happening there. So compare cytokinesis in a plant cell and in an animal cell. So if you don't remember that, remember one of the cell types has a cell plate and another cell type has a cleavage furrow. So you tell me, which one is which? You should say that the cleavage furrow is unique to the animal cells. Okay, and that's where the cytoplasm kind of like pinches in. Okay, right here. And that's referred to as the cleavage furrow. In a plant cell, you have a cell plate. And that's important because it goes on to form the cell wall. All right. Now let's look at the second question. Which stage of the cell cycle does a chromosome consist of 2 identical chromatids, sometimes referred to as sister chromatids? So sister chromatids are first formed after the DNA or after the chromosome has been replicated. So that starts in S phase, okay, which is in interphase. Specifically S phase. That's when the DNA is synthesized. And then the chromatids don't exist anymore once each cell has its own chromosome. Okay, so after they split, then they're no longer chromatids, they're considered chromosomes. So remember, at the beginning in interphase, you have one chromosome that looks kind of like this. And then once those chromosomes replicate, you have your sister chromatids, or like that. And so when those pull apart and separate, then they're not considered chromatids anymore. And so that happens in anaphase. So sister chromatids exist from S phase of interphase to anaphase when they pull apart. Okay? So that one was a little bit trickier. So let's go on with the control of the cell cycle. So today's lesson is pretty detailed. It's got a lot of extraneous information, I'm just going to go ahead and say. But I want you to kind of get the big picture here, okay? I want you to see that the cell cycle is very tightly controlled. I want you to walk away from this lesson understanding that all this stuff doesn't just happen by chance, that there's a lot of signals that are happen and stops and starts and regulation and it's, a machine or a factory almost working in there and making sure that everything happens just so. And that happens at the tiniest cellular level, to be able to control the process of cell division. And when that's not controlled, then you have things like cancer, okay? And we'll talk about cancer more at the end of this lesson. So, cells seem to know what stage they're in. There's molecules that are present in them, in the cytoplasm, that cause them to divide or cause them to stop. The cell cycle has checkpoints in it. So we see major checkpoints at G1, G2, and M, okay? And if those cells, at those points, if the cell doesn't meet a certain criteria, then it's halted. Cell division is stopped, okay? It can't move on if it doesn't, check the list off there at the checkpoint. Okay, and so what we see, the G1 checkpoint is thought to be perhaps the most important checkpoint. Generally, if a cell makes it past the G1 checkpoint, it will go ahead and complete the rest of the cell cycle. Okay, now there's one other phase that we have not talked about, and it's referred to as G0. That little zero down there is not, I don't know what it is. It's not a Greek letter, but I don't know what it is. But G0 is a non-dividing cellular stage. So when cells are not in interphase, they're not growing and not dividing, then they're considered to be at rest. They're in a G0 stage. And so if the cells fail to meet these checkpoints, then they are sent back to G0, okay, until they can do whatever they need to be able to pass the test, okay? So it's also important to know that cells divide at different rates. And so some cells are going to maintain in G0 forever. For example, brain cells. If you kill your brain cells, you're not getting any more. How many you have now is how many you'll have for the rest of your life. Now you can make more connections between brain cells, and that's kind of how we get smarter and use a larger capacity of our brain. But you don't actually get more brain cells. It doesn't divide anymore. And so those cells are constantly in a G0 stage. And so different cells divide at different rates. So in the cell, there's all these cellular surveillance systems that make sure that things are present before the next step goes on. You see this too in like a washing machine. For example, when you start a load of clothes in your washing machine, if you have a newer washing machine, it may tell you on the thing, it's going to take 42 minutes or whatever. Now, if you time it, it may take longer than 42 minutes. But that's just an estimation based on how long it normally takes to get the checkpoints, right? And there's certain things that have to happen. So before the washer will actually start dispensing the soap, it has to have the water in it, right? So the water fills up, and before it dispenses the soap, then the water has to be in it. And then before it starts spinning, then the soap has to be in it. And then, before it stops, it has to drain. And so there's, just like there's checkpoints there, in that cycle, there's checkpoints in the cell. So if you kind of think about it like that, then it may be a little bit easier to manage. So in the cell cycle, there are two main regulatory proteins, and these should be somewhat familiar to you. So the first is protein kinase. If you remember, we talked about protein kinase before. Its major function was phosphorylation. Okay. And so phosphorylation means to add a P. And generally we saw that would activate other molecules in the cell signaling pathway. Okay. Now, these proteins are generally present in constant amounts in the cell. They don't fluctuate over time. They're present. They may be inactive, but they're still there. Okay. Another regulatory protein that we have in the cell are cyclins. And they are cyclic, meaning they fluctuate in concentration in the cell. They, there's a lot and then there's none, and then there's a lot and then there's none, and then there's a lot. And so what these do is they bind to and activate protein kinase. Well, you may say, Ms. Brown, I've never heard of a cyclin before. I thought you said these were familiar. Yes, you have. You have heard of a cyclin before, and it was called CAMP. If you remember, CAMP stood for cyclic AMP, okay? And it's cyclic because it's a cyclin. It also fluctuates in concentration in the cell, okay? And it activates protein kinase, okay? Once it activates protein kinase, then protein kinase can phosphorylate these other molecules that help to start the cell cycle. So, CAMP, when we talked about it before, we talked about it as a second messenger, right? So you had, a G protein coupled receptor or some type of cell receptor, and it received a signaling molecule, okay? And that signaling molecule would activate, something like adenyl cyclase, which activated CAMP, our second messenger here, our cyclic AMP, which would activate protein kinases, which would have some type of response, such as starting the cell cycle, okay? And so this is kind of some application to what we've been talking about all the way along. All right, so, and then how do we turn this off? If we want to turn this process off, well, we can do that by dephosphorylation, which is the enzyme protein phosphatase, okay? That would help to turn this off, all right? And so when these cyclins accumulate, then that's gonna, they're important because they can help trigger cells to go in past a checkpoint, okay? So what specifically that we are looking at here, the specific type of kinase that we are looking at here is a class of kinases called cyclin-dependent kinases, or CDKs, okay? So you have your protein kinase, your cyclin-dependent kinase, or CDK, which binds to a cyclin once they've accumulated, okay? Because again, they're cyclic. They rise and fall and rise and fall and rise and fall. And when those two come together, they create maturation promoting factor called MPF, or sometimes it's referred to as M phase promoting factor. Once that's formed, then that meets the requirement for the G2 checkpoint, and it allows it to go on into M phase, which is why it's sometimes referred to as M phase promoting factor. M phase promoting factor, okay? At the end of M phase, those cyclins will be degraded, okay? And then, so the concentration of it will drop back down like we see in the graph here. And then that CDK will be accumulated back, right? And allowed to build up for the next round of the cycle, okay? We see this graph of it right here. As the cyclin concentration goes up, then the MPF activity spikes because those two are dependent on one another. You have to have cyclins to make the MPF, okay? And we see that here in this graph. You see them really spike at M phase, and that's when you have the M phase promoting factor, okay? So cyclins plus CDK equals the maturation or the M phase promoting factor. The activity of that is directly proportional to the concentration of cyclins, like we just said. And so it goes on to phosphorylate many proteins, which initiates mitosis, all right? And that's really important. Now, other regulatory molecules that we have in the cell are growth factors. And so growth factors are proteins that are released by other cells that causes cells to divide. And we'll talk about those in just a second. But again, back to these checkpoints. This was more of an example of an external cell signaling, okay? this more an example of an internal cell signaling. All right. We know of over like 50 different growth factors that cause the cell to be able to respond to. And they can occur in different combinations to produce different signals. But back to these checkpoints. So, for example, there's a checkpoint in anaphase. If you think what happens in anaphase is when the chromosomes pull apart. Okay? What needs to happen? What do you have to make sure that happens before all the chromosomes pull apart? Well, first, you want to make sure that the chromosomes are attached to a spindle fiber. Okay? That every sister chromatid is attached to a spindle fiber. Because if they're not attached to the spindle fiber, remember the spindle fibers are like the little arms that connect to them, right? That help to pull them apart, okay? And so if they're not attached to the spindle fiber, then when those cells separate and divide down the middle, then you would have some in pairs, right? That they don't get pulled in the right way. And that would cause an irregular number of chromosomes. We see that in when it happens in sex cells. We see that, and it's called Down syndrome. You have an irregular number of chromosomes. And so there are regulation molecules and signaling molecules that tell a cell, hey, don't pull apart yet. We don't have everybody lined up. We don't have everybody all in the right place. All the spindle fibers aren't attached to the centromere. And for anaphase to begin. Okay, so when cells lose the ability to control the cell cycle, that results in cancer. Okay, cancer cells have lost the ability to control that. So cancer cells don't stop dividing when growth factors are depleted or when it ignores those checkpoints and those signaling molecules that come from the cell, and they continue to divide forever so long as nutrients are present. So, for example, there are a set of cells called the HeLa cells that have been reproducing in culture since like the 50s. They came from an African American lady named Henrietta Lacks. I'm trying to look at the 1951. I think, came from Henrietta Lacks, and they were kind of taken from her really without consent, and the science community has profited a lot of money on these HeLa cells. You can go to a biological supply company. I could go online and order a sample of these HeLa cells for us to do scientific experiment with if we would want to. But they came from her tumor, and they continue to reproduce. As long as they have a nutrient broth to grow in, they will reproduce infinitely. And they they reproduce more rapidly, and they just ignore all of these normal cell cycle controls. So some of the controls that we normally have that prevent cancer are density-dependent inhibition and anchorage dependence. So anchorage dependence means that cells will normally need some type of substrate to grow on. And so they receive signals from the plasma membrane of proteins that allow them to know that they can grow in that space. And so that's referred to as anchorage dependence. Density-dependent inhibition means that the cells like to grow in a nice thin sheet until they fill the space, and then they stop. If the cells get damaged or scraped out, removed, then they'll replace those cells, and then they'll stop again. So we see that in normal cells. But in cancerous cells, we don't see that. And so what we see is these cells don't really exhibit these cell cycle controls. They ignore these signals. And so they will just divide and divide and divide. And ignore that. This is another picture that just kind of shows that density-dependent inhibition. And I believe I talked to you all about that once before. So normally, even in the event that the cells ignore these signals. The body's immune system can often recognize a cell that is ignoring all these signals and destroy it. But sometimes cancer cells can also manage to evade that destruction as well. And so when a normal cell becomes a cancerous cell, this process is known as transformation. Cancer can be benign or malignant. So benign tumors are just going to kind of stay in one area. They're generally not going to cause a lot of problems. They can go in and cut them out and remove them and it not be too bad. Now, malignant tumors are the more negative kind that tend to spread to new tissues and cause problems in more organs. And that process is called metastasis. When cancer cells spread to new locations, that's referred to as metastasis. And so here we see a tumor that's in the breast tissue, and you see it's starting to grow, okay, getting bigger and bigger and invading foreign cells. Now, some of these cells then, if they break off and they get loose into the blood vessel, then they can move to other parts of the body and find somewhere else to anchor, or maybe really even not anchor, maybe just travel throughout the blood. And so that is a metastatic tumor then when it finds a new location that's secondary to the original source. Okay, cancer. And so they are looking for cells that are in mitosis. And so the way that they act are Taxol, for example, is a drug that's often used for chemotherapy, freezes the spindle fibers in mitosis. So if you remember, the spindle fibers are what attach to the sister chromatids to be able to pull it apart and separate it. And so if those spindle fibers are frozen, then those cells can't separate, those cells can't divide. And so, mitosis is, like I said, a pretty simple process, but knowing it and understanding it is really important to be able to help with cancer treatment, because that's what we're targeting. We're trying to stop the cell cycle and stop cell division. So let's go ahead and look at the assignment for today. So today you're doing this mitosis in cancer cells. And it should be pretty straightforward. You'll want to read just the little intro here. But you have two tables of mitosis in normal chicken cells in table 1 and then in cancerous chicken cells in table 2. So both of them, basically what they did is similar to the way that you did it the other day. You counted 100 cells, they counted 500 cells, and looked at the different phases of mitosis. So 500 cancerous cells, 500 non-cancerous cells. And then they looked at the time that it took for these cells to complete mitosis. So notice here in the normal cells and in the cancerous cells, the cancerous cells took 448 minutes versus the normal cells took 625 minutes. So the cancerous cells are only like 2/3 of the amount of time. So that's pretty crazy. So remember, because they evade some of these cell cycle controls, they can go through the process a lot more rapidly, and that's not necessarily a good thing. So what you have to do here is you have to fill out the rest of the table. So you'll have to figure out what percent of the time they spent in, you'll do this column first, okay? And so you'll do 424 out of 500. And then 424 out of 500 and get it as a percent. And then what percent that is going to be. 50 out of 500, and that would be 10%. Okay, and so on and so forth. And then you'll multiply those percentages times the total amount of times to get your minutes that it would take. Okay, and so that one was easy to do because it was 10%. And so you'll figure out the minutes for each one. So that kind of describes step 1 and 2. Now in step 3, it says prepare a graph to show these results. So what kind of graph do you think that you're going to use? You might naturally think bar graph because Ms. Brown says, or line graph because Ms. Brown says we like to use line graphs in this class, but that is actually going to be wrong here. You may also say pie graph because we're looking at 100%. That's also going to be wrong. It's going to be a bar graph because we are comparing two sets of values. So you'll have your phases on the bottom here, and then you'll have a cancer cell and a normal cell, a cancer cell and a normal cell, a cancer cell and a normal cell. Make sure to give me a legend to show me which one is a cancer cell and which one is a normal cell. You may want to color code those. Give me a label. Give me a label over here and a title on your graph. That's all very important when you make a graph. If you have graph paper to do that on, that's great. If not, just do it on regular notebook paper. There's some blank space at the bottom of the back that you can put it on. And then you have just 3 little questions to answer. So this is important about understanding cancer cells and mitosis in cancer cells. So y'all let me know if you have any questions on that. You have a test coming up on Wednesday, so you've also got a study guide that you can begin to work on today. And then tomorrow we'll do the personal progress check. And there's an extra credit lab. If you want more information on that, come in today and pick up your materials or email me if you have questions. Y'all have a great day.